Mesenchyme is a tissue of the embryo. It's embryonic tissue which consists of undifferentiated mesenchymal cells. These cells differentiate and give rise to all types of connective tissue, including bone and cartilage. So all these tissues are mesenchymal tissues. This is connective tissue. It consists of three components: cells, extracellular material, which is composed itself, fibers, and ground substance. So cells, fibers, and ground substance are components of connective tissue. As you can see, there is large amount of extracellular matrix in this tissue, and cells are surrounded by matrix. So unlike epithelial tissue, which is composed mostly of cells, this tissue contains great amount of extracellular matrix, and cells are not adhere to each other. They don't form continuous layers. There are three main types of fibers in this tissue: collagen fibers, elastic fibers, and reticular fibers. And there are different macromolecules in the ground substance, but we will discuss this next lesson. Today we will talk only about cells. All components of matrix, proteins, glycoproteins, glucose aminoglycans, are produced by the cell, which is called fibroblast. Now you can observe here transmission electron micrograph. This cell is fibroblast. It is surrounded by extracellular matrix. You can see cross sections and longitudinal sections of collagen fibers in the matrix. Collagen fibers are made of protein collagen, elastin, and other proteins are produced by fibroblasts. So this cell is synthetically very active. That's why it has very well developed rough endoplasmic reticulum. You can observe this organelle in the cytoplasm, and very well developed Golgi apparatus. It has euchromatic nucleus. You can see here euchromatic nucleus with prominent nucleolus. Everything indicates high level of synthetic activity. Shape is irregular. This cell has many processes. You can ensure this is fluorescent microscopy. So this cell is very active. It secretes collagen, elastin, multi-adhesive glycoproteins, proteoglycans, all components which occur in the extracellular matrix. The quiescent form is called fibrocyte. So this is the same cell but with low level of synthetic activity, and it differs morphologically from active cell. It is smaller, it has less processes, it is spindle shaped, it has less developed rough endoplasmic reticulum, Golgi apparatus, and it has more darkly stained nucleus because nucleus is heterochromatic. So fibroblasts and fibrocytes are most numerous cells in connective tissue. They are permanent residents of this tissue. They originate locally from the mesenchymal cells. Is it clear? If something is not clear, ask me right now. Is it clear for you?
Yes,
Yes, doctor.
Yes, doctor.
This cell is called macrophage. It is monocyte derived cell. What I mean, bone marrow contains stem cells that give rise to blood cells, including monocytes. So stem cells, which are found in the bone marrow, give rise to monocytes. Monocytes circulate in the blood, but monocytes can cross the wall of blood vessels and penetrate connective tissue. In the connective tissue, they differentiate into macrophages. That's why we can say that macrophages are monocyte-derived cells. What is characteristic for these cells? Phagocytic activity. They can engulf dead cells, bacteria, and other invaders. They can engulf components of extracellular matrix. Do you remember what is phagocytosis? When
Yes,
what is
teacher,
phagocytosis?
when we engulf large materials to inside the cell.
And what happens then with phagosome? Phagosome fuses with lysosome, with
Yes,
primary
it
lysosome to
makes it
form
light in those
secondary
light lysosome.
lysosome. We studied this during cytology. So phagosomes fuse with primary lysosomes to form secondary lysosomes, and in secondary lysosomes, digestion of engulfed material takes place. So that's why the cytoplasm of this cell contains many primary and secondary lysosomes, as well as phagosomes. You can see these organelles in the cytoplasm of the macrophage. This is activated macrophage. It has very irregular shape with many protrusions and invaginations that increase surface area. Next type of cell, mast cell or tissue basophils. They also have precursor cells in the bone marrow. Their cytoplasm is filled with secretory granules, and sometimes these granules cover the nucleus, and nucleus is not visible very well. This cell is ovoid or rounded. It has rounded nucleus, which is located centrally, and you can observe heterogeneous granules in the cytoplasm. These granules stain with basic dyes, so they are basophilic. But pay attention that they can change the color of dyes. For example, if we use for staining blue dye, toluidine blue, granules change the color of this dye from blue to red or purple. This is called metachromasia. And metachromasia occurs due to high content of sulfated GAGs in the granules. These granules contain heparin, and heparin interacts with dye and changes the color. Uh, Not only heparin is inside the granules. This cell produces and stores several types of biologically active substances. For example, histamine, which increases permeability of blood vessels, heparin, which is anticoagulant, enzymes, which are which are called serine proteases, chemotactic factors that attract eosinophils and neutrophils. These are all released outside the cell when cell receives a payment from the appropriated stimulus, and this is called plasma cell. It is a very important cell because it produces antibodies. Antibodies are produced against antigens called immunoglobulins. They are glycosylated proteins and they are produced on RER and then glycosylated in the Golgi apparatus. That's why this cell contains very well developed RER and Golgi apparatus. You can observe that the cytoplasm is filled with cistern of area. That's why the cytoplasm is basophilic. You can see the light microscopy image of plasma cell. It is a large spherical cell with a rounded nucleus. Nucleus is located eccentrically. It means not in the middle of the cell, but eccentrically. The cytoplasm is basophilic because it is filled with cistern of Rath and opposing reticulum. But this small region, note this small region next to the nucleus, it is pale. Why? Because it contains Golgi apparatus and centrioles. So Golgi apparatus and centrioles are located near the nucleus, and that's why this region appears pale. Nucleus has clock face appearance. Why? Because of composition of euchromatin and heterochromatin. If you remember, euchromatin is light stained and heterochromatin is more electron dense, and alternating regions of light and dark euchromatin and heterochromatin regions gives a nucleus clock face appearance. These are morphologic and staining properties of this cell that help you to identify this cell. So as I told you, plasma cells produce antibodies. Plasma cells are derived from B lymphocytes. B lymphocytes are leukocytes, blood cells, and they have precursor cells in the bone marrow. Now look at this tissue preparation. These large spherical cells are called adipocytes or fat cells. They occur in loosened, dense connective tissue singly or as a small groups. But
Doctor,
sometimes what?
doctor, can you repeat what are they called?
Adipocytes or fat cells because they are specialized for lipid storage. They accumulate neutral fats. These cells are large spherical cells. Nucleus is always at the periphery because the cytoplasm contains one large droplet, lipid droplet, which contains triglycerides. So this cell is specialized for lipid storage. And large aggregations of adipocytes form adipose tissue. Adipose tissue is special type of connective tissue and it's described in your textbook. We will describe this tissue in details later. But these cells, adipocytes, occur in the connective tissue as well. They originate locally from mesenchymal cells. They are permanent residents of connective tissue. In addition to these cells, which I already have described, you can find blood cells in the connective tissue. Because, as you know, blood vessels are always surrounded with connective tissue, and blood cells, leukocytes, not erythrocytes, RBCs, but leukocytes can cross the endothelial wall of small blood vessels and penetrate connective tissue, and they make population of wandering cells in connective tissue. So some cells in connective tissue are permanent residents and some cells are moving here from the blood. Now I will list once again all cells you should know for next lesson. Fibroblasts and fibrocytes. They have different level of synthetic activity and that's why they differ morphologically. And as I told you, fibroblasts secrete all components of extracellular matrix. Macrophages, they are monocyte-derived cells, and in addition, I want to tell you that macrophages of connective tissue are also called histiocytes, but you can find monocyte-derived cells in many organs, but in different organs they have different names. We will study bone tissue soon, and you will see that bone tissue contains cells which are called osteoclasts, and they are also monocyte-derived cells. Next semester, you will study skin, respiratory system, liver. In liver, you can also find monocyte-derived cells. They are called Kupffer cells. In the skin, they are called Langerhans cells. In the nerve system, they are called microglial cells. So different organs and tissues contain monocyte-derived cells with different names, and all these cells comprise a large cellular family, which is called mononuclear phagocyte system. All these cells are derived from